Hello, and welcome to COVID-19, What Pharmacists Know Now, a series brought to you by the Canadian Society of Hospital Pharmacists, BC branch. Today's topic is Injecting Knowledge, a COVID-19 vaccine update. My name is Jillian Reardon, and I'm a clinical pharmacist and lecturer with the UBC Pharmacist Clinic, Faculty of Pharmaceutical Sciences at the University of British Columbia in Vancouver, Canada. I have no relevant conflicts of interest to disclose, and will make the disclaimer that because the COVID-19 pandemic is a rapidly evolving situation, there are many unknowns with limited research and knowledge changing daily. I think it's safe to say we have more questions than answers, especially when it comes to a vaccine. The content of this presentation can be considered up to date as of April 5th, 2020. In this presentation, we will review vaccine basics, including how they work and the steps in vaccine development. Considerations for creation of a COVID-19 vaccine will be discussed with a look at what research is happening globally. Finally, the process by which vaccines may be regulated in Canada during pandemic situations will be explored. We have seen a very rapid progression of COVID-19 from the first cluster of patients in December 2019 to in January, Chinese researchers isolating and sequencing the genetics of the novel severe acute respiratory syndrome coronavirus 2, or SARS-CoV-2, the virus responsible for causing COVID-19, thus laying the foundation for vaccine development to commence. On March 11th, the World Health Organization declared COVID-19 a pandemic, further fueling the urgency to develop a vaccine. While no proven vaccine currently exists for COVID-19, the WHO estimates that 52 vaccine candidates are being developed and tested by companies and academic institutions around the world. Globally, at least 4 research groups have already started animal testing, and 3 research groups have started or will soon start human testing. In Canada, Justin Trudeau announced $192 million to fund COVID-19 vaccine development and emphasized Canada's readiness to mass produce a vaccine, regardless of who creates it first. So why is so much emphasis being put on the need for a COVID-19 vaccine? Well, effective vaccines not only protect those immunized, but can also reduce disease among unimmunized individuals through herd immunity. Herd immunity is achieved when a large percentage of people in a population are immune to a pathogen. Herd immunity can only be achieved through natural infection or vaccination, with vaccination being the obvious preferred choice. In the absence of vaccination or other precautions, pathogens will spread quickly and kill people, while only the recovered will be immune. With implementation of physical or social distancing, many more will survive as less people will be infected. Those that are infected and survive will become immune. If an effective vaccine is developed and deployed, most will survive and become immune, thus dramatically slowing viral spread and boosting herd immunity. The bottom line is with COVID-19, the only safe way to achieve herd immunity is through vaccination. It is estimated that vaccines prevent almost 6 million deaths annually worldwide. As far as public health interventions go, only clean water performs better. As a result of widespread vaccine use, the smallpox virus has been completely eradicated, and the incidence of polio, measles, and other childhood diseases has been drastically reduced around the world. This table illustrates the effectiveness of vaccination by comparing the number of cases of 6 vaccine-preventable diseases in Canada before and after the introduction of vaccines. Of note, this data is from 2011 to 2015, and in more recent years, there have unfortunately been local and global outbreaks of vaccine-preventable diseases. A recent notable example being measles due to suboptimal vaccination and population immunity gaps. A vaccine works by presenting certain molecules, also known as antigens, of the virus to the immune system. Traditionally, these antigens are a weakened or an active form of the virus, so they can't actually cause the illness. Our immune system is then able to recognize the antigen as an unwanted foreign invader and forms antibodies, allowing the body to remember the pathogen in case it tries to infect you in the future. At which point, the antibodies you've built up will then quickly attack the virus before it is able to make you unwell. Newer technology is now also allowing creation of vaccines that are not made from the weakened or inactive pathogen at all, but instead from a copied genetic code from the virus, which is made in a laboratory. More on this later. Let's look now at what factors need to be considered in development of a COVID-19 specific vaccine. Human coronaviruses were first identified in the mid-1960s, and there are 4 that commonly infect humans, causing self-limiting common colds. These are believed to have evolved in humans to maximize their own spread, which means sickening but not killing people. Sometimes coronaviruses that infect animals can evolve to become a new human coronavirus. Examples of this are SARS and MERS coronaviruses, and now SARS-CoV-2, causing COVID-19 disease. Coronaviruses are named for the crown-like spikes on their surface. There are 3 proteins present on the viral surface, the envelope, membrane, and spike, which all encapsulate a strand of RNA. This RNA molecule holds the genetic instructions that make up the virus. These proteins and genetic material all represent possible targets for vaccine development. Unfortunately, no safe and effective vaccines against coronaviruses have been developed. Decades ago, a vaccine for a coronavirus in cats, known to cause feline infectious peritonitis, was developed, however actually ended up increasing the chance of cats getting sick with the virus. Similar phenomenon were observed in animal trials of SARS vaccines. With both SARS and MERS, extensive research into vaccine development did occur, and some success in animal models was found. However, epidemics ended before vaccine development was complete, thus halting research. As SARS-CoV-2 shares between 80 and 90% of its genetic material with the virus that causes SARS, researchers have now resurrected previous vaccine work, allowing for very rapid development of COVID-19 vaccine prototypes. Research indicates SARS-CoV-2 changes slowly and doesn't appear particularly good at dodging the immune system, which provides hope that an effective vaccine will be found. Viral vaccines on the market today use one of several approaches. Live vaccines use a weakened or attenuated form of the virus. An example of a live vaccine is measles, mumps, rubella. Inactivated vaccines use a killed version of the virus, such as with the annual flu shot. Subunit, recombinant, polysaccharide, and conjugate vaccines use specific pieces of the virus, like its protein, sugar, or a casing, to elicit an immune response. Examples of these types of vaccines include hepatitis B and HPV. Conventional vaccine approaches such as these provide durable protection against a variety of dangerous diseases. However, it's time and labor intensive to grow viruses in facilities which can guarantee their purity. Performing this process on a pharmaceutical grade scale can take years. In a pandemic situation, there is a need for more rapid development and large-scale deployment of vaccine. Because of this, some researchers are employing new technology using viral RNA or DNA. In fact, one RNA-based vaccine for COVID-19 has already entered human trials. Unlike traditional vaccines, where the killed virus or part of the virus is directly introduced to generate an immune response, RNA-based vaccines give the body the molecular instructions or blueprint in the form of messenger RNA to make the viral protein itself. This protein gives the immune system a preview of the virus so it can be recognized and neutralized if exposure to the real virus does occur. DNA and RNA viruses are in theory faster and cheaper to produce than traditional vaccines. They can be made quickly because they don't require culture of viruses and instead use synthetic processes that can be standardized and scaled up to facilitate quick responses to large outbreaks and epidemics. So when can we expect to have a vaccine against COVID-19? Traditionally, vaccine development and testing occurs sequentially and often takes 10 years or more. Once animal testing is complete, clinical trials in humans can commence, which consist of 3 phases. Phase 1 involves a few dozen healthy volunteers and tests the vaccine for side effects. Phase 2 involves several hundred people, usually in part of the world affected by the disease, and studies if the vaccine is effective. Phase 3 is a much larger trial, usually several thousand people, who will be randomly assigned to treatment with vaccine or placebo to give a more robust measure of vaccine effectiveness and safety. In a pandemic situation, multiple activities related to vaccine development and testing must occur concurrently. Some steps may be expedited or skipped entirely, with some scientists arguing the risk of delaying the advancement of vaccines is much higher than the risk of causing illness in healthy volunteers. Very early manufacturing and scale-up to commercial capacity must occur before safety and effectiveness is established to ensure that if vaccines are successful in human trials, they can be distributed as quickly as possible. In the case of H1N1 influenza pandemic, a vaccine was developed relatively rapidly, largely because influenza vaccine technology and manufacturing was already very well developed. Despite the steps being taken to expedite development, scientists estimate it will be at least 12 to 18 months before a COVID-19 vaccine is widely available. And that's assuming there's no bumps along the way, such as side effects, dosing issues, and manufacturing problems. This table summarizes the various COVID-19 vaccine technologies under investigation. As you can see, 2 vaccines have started phase 1 clinical trials in humans. Massachusetts-based Moderna, with their new mRNA based platform, and China's CanSino Biologics with a non-replicating vector technology. Other phase 1 trials are expected to start later this month. Now you may look at this table and ask, shouldn't everyone focus their efforts into creating a single vaccine? It's impossible to anticipate how a vaccine will perform in human trials, so diversity is key to increase the likelihood of finding a vaccine that's safe and effective. Furthermore, as many new vaccine technologies are being tested in the development of a COVID-19 vaccine, it's unknown if these will be scalable. On March 16th, the NIH announced that the first participant entered its phase 1 study for a vaccine called mRNA 1273, a mere 63 days from viral sequencing to first human dosing. This phase 1 study will provide important data on the safety and efficacy of different doses of mRNA 1273. The open-label trial is expected to enroll 45 healthy adults aged 18 to 55 over approximately 6 weeks. Study participants will receive 2 doses of the vaccine via injection in the upper arm, approximately 28 days apart. Participants will be monitored for side effects and response for up to a year. As it's early days in the search for a vaccine against COVID-19, many unanswered questions and challenges lie ahead. Conducting clinical trials during a pandemic poses additional challenges as it's difficult to predict where and when outbreaks will occur and to prepare trial sites to coincide with vaccine readiness for testing. Given the public fear and uncertainty, as we progress to phase 2 and 3 trials, people may be reluctant to enroll in a study where there's a chance they'll receive placebo and not vaccine. For novel vaccine technologies, most of which are unlicensed, large-scale manufacturing has never been done. Furthermore, once we have a vaccine, how effective will it be? The overall effectiveness of vaccines is influenced not just by the vaccine's ability to stimulate an immune response, but also the degree of public uptake, as well as timing of vaccine availability in relation to pandemic activity. As with naturally acquired infection, the potential duration of vaccine immunity is unknown. Additionally, the virus causing COVID-19 could eventually mutate in ways that would make previously effective vaccines ineffective. Because pandemics are a global threat, there are significant ethical considerations regarding Canada's responsibilities to the global community, such as international cooperation and assistance to other countries to promote equitable access to vaccines. Let's look now to the Canadian context. Health Canada is the regulatory authority in Canada responsible for ensuring the quality, safety, and efficacy of all biologic drugs, including vaccines. Health Canada has developed a Canadian pandemic vaccine strategy, which is informed by lessons learned during H1N1. Normally, before a vaccine can be submitted to Health Canada for approval, sufficient scientific evidence must be collected to show that it is safe, efficacious, and of suitable quality. This scientific evidence includes results from human clinical trials. For a vaccine to be useful in mitigating the pandemic impact, it will need to be available almost immediately after it is manufactured. Therefore, standard regulatory processes do not apply. Under Canada's pandemic vaccine strategy, regulatory approval of a vaccine can occur by various pathways. If the pandemic is caused by influenza, viral strains change, but the vaccine technology remains the same, allowing for prior authorization based on limited data. In non-influenza pandemics, such as what we're facing now, where phase 3 clinical trial data is available, the usual review process can be fast-tracked, with approval granted to vaccine manufacturers based on assurance that they will provide a rigorous post-market safety and monitoring plan. When less data is available, the extraordinary use of new drugs regulations, enacted in 2011 after H1N1, can allow Health Canada to expedite authorization of a pandemic vaccine based on animal data, supplemented by any available human data, as long as complete manufacturing information is available and a rigorous post-market surveillance plan is in place. Finally, as a last resort, an interim order based on a state of public health emergency may be issued by the Federal Minister of Health. Such an interim order can suspend certain requirements of the Food and Drug Acts, allowing the issuance of a time-limited authorization with additional manufacturer obligations. As initial supplies will not be sufficient for everyone in Canada, the National Advisory Committee on Immunization will develop recommendations for the prioritized use of vaccine to ensure those at highest risk receive the vaccine first. Finally, a common question that comes up is, will pneumonia or flu vaccines protect against COVID-19? The answer is no. This is a novel virus which requires its own vaccine. However, all individuals are encouraged to follow public health advice for recommended vaccines to protect against other respiratory illness and communicable disease. In closing, although promising research is ongoing, there is currently no vaccine for COVID-19 disease. Until a vaccine and/or other treatments are developed, our best protection remains exercising preventative measures through physical distancing and proper hand washing. You can find more information about the COVID-19 pandemic on the Canadian Public Health Agency and BC Centre for Disease Control websites. Additionally, for more general information on vaccines, you can consult the BC immunization guides from the BC CDC or Immunize Canada. Thank you so much for listening and stay safe.